Can you hear me? May I start? May I start the lecture eventually? Okay. So, come on, let's go. Let's do this. Just a few quick announcements because next week I'm not teaching, but we're going to have a review session on Wednesday nonetheless, only with me because this coming midterm is only going to be about lectures I taught, including this one. And that's, so someone was asking me, it's cumulative in the sense that I don't want to be in a position where I'm going to ask you a question and it refers to some knowledge from the previous lectures, and so you would come to me and say, oh, but that was not on the exam, right? So in this regard, it is cumulative, but in terms of content, there's a greater emphasis on the lectures that we've covered since the previous exam. Okay, does that make sense? So in other words, there might, I expect you to not have just completely forgotten what we talked about in the lead up to that previous exam. And so in this regard, because there's an exam, and so although I will have a review session, and although I'm not teaching, I'm still going to have an office hour on Tuesday, but I can't have any on Thursday because I'm giving a talk elsewhere in New York. So, and as that's going to be a difference from last time where I feel I answered questions all the way up to the last minute, including by email. I won't be able to do that next week. So, and I'm saying it on record purposefully, basically the review session is the deadline to ask me anything, okay? Because I won't be available on Thursday and in the lead up to the exam next week. So just be prepared for that. And I still have to post some homework to help you study, which I'll do today. And with no further ado, now we're going to talk about polarities and asymmetries. All right. So before I jump and comment on this first slide, I just want to say that everything I told you so far, whether discussing the secretory pathway or the cytoskeleton and the dynamics, we're at a certain level of organization, very molecular, very specific here with polarity. And in the next lecture, which is going to be, I think, in 2 or 3 weeks from now, 2 weeks and a half, with migration, we're going to discuss more integrated processes. And so one of the things I'm going to try to do is help you guys see the connections between this and the different lectures we've talked about and see how everything is connected and part of more integrated processes. So here to start illustrating this concept of cellular polarities and cellular asymmetries, which is, the two terms are used in somewhat synonymous ways, right? So the first example here are epithelial cells, and we've talked about those. And I've emphasized how there's an apical surface. So if we focus on this first one here, the asymmetry or the polarity is very clear, right? You have this side, let's say, so if this is the gut epithelium, that side faces the lumen, so that's the outside of the body, strictly speaking. Although it's inside, I know, but it's actually outside. Does that make sense? And then, so this is the first layer. There's a very tight barrier that separates the apical surface and the basolateral surface, which then contacts this connective tissue and by extension the blood and the immune system and so on and so forth. Okay, so you have, for the function of the enterocytes of the gut, the digestive function, you have this need for polarized cells. Okay, do you remember how these so if now I'm asking you, what, in molecular terms, what does that mean to be polarized? What are in membranes? What kinds of molecules are in membranes? Proteins, phospholipids. Good. Okay, now I'm telling you there's a difference between the apical side and the basolateral side. What kind of differences do you expect then?
Proteins and lipids.
Yeah, so certain proteins, different kinds of proteins, different kinds of lipids on either side, right? That's fairly simple. Now, I've told you this. How is that achieved? How do you position different proteins at the membrane on the apical side versus the basolateral side? Vesicles, right? So this is the kind of, yes? Kind of a pH. Well, so the pH, there's a connection with the pH, a remote connection, because there's a gradient of pH in the secretory pathway, right? But the kind of knowledge and the kind of connections I want you to be able to make, and I don't want you to forget from one exam to the next, is although the dispatching of proteins in separate vesicles happens in sorting compartments in the trans-Golgi network, and those vesicles are fated to go to the apical membrane or the basolateral membrane, okay? So this is very important for polarity. Okay, so that's in this regard. If I want to ask you questions about polarity in the upcoming exam, I feel entitled to, I expect you to remember this sorting business. Okay, that was my point about the cumulative. Okay, now this was for epithelial cells, so all kinds of polarity, and I'm not going to comment on the different kinds of epithelia at the tissue level. There are many different layers, but migratory cells, more mesenchymal cells. And you remember the key difference between epithelial cells and mesenchymal cells? Come on, give it to me. The difference between epithelial cells and mesenchymal cells. Yes? Yes, that's right. So it's epithelial cells are engaged primarily in cell-cell adhesion, right? Epithelia is a bunch of cells together. Mesenchymal cells, they have more interaction with the extracellular matrix, right? So they are part of so-called connective tissue, where there's an abundant extracellular matrix. And the most compelling connective tissue in this regard is the blood, okay? Because in the blood you have individual cells bathing literally in a huge matrix, which is the plasma, okay? So going back to this migratory cell, here, if this cell is migrating in this direction, polarity is obvious, right? So it jumps at you. You always have, or often have, like in this case, a very conspicuous lamellipodium. That's a lot of protrusive activity, a lot of actin dynamics, right? So again, there are connections to be made between this behavior and all the actin dynamics we've talked about two lectures ago. So this is often called the leading edge. There are also microtubules organized in a particular way. And at the trailing edge, instead, the cell is sort of constricting and moving forward. So constriction, what kind of protein would you want to invoke for constrictions? Dynamines could be, but let's say actin-based. Myosines, right? So myosin, and we'll see that when we talk about the migration lecture, there tend to be polarized activities of certain Rho GTPases like RAC, CDC42, especially RAC, which promote the formation of the lamellipodium in the front, versus Rho, which tends to promote contractility in the back. So when we want to discuss and think about polarities and asymmetries, there are a few points that we need to keep in mind, and I'm going to highlight them here. Sorry, there's more text on that slide than I like, but really this boils down to these 3 points I'm going to touch upon today, and also quite a bit in the migratory lecture, the lecture on migration. So the first one, we've already started to discuss that. What do we mean when we talk about polarity, when we talk about an asymmetry? In other words, what are the components that are not evenly distributed? Okay, so we're going to have to think about that and see how that matters. And then from that standpoint, you can imagine, you can look in two directions. One of them is, How is this established? Where does that come from? And often cells polarize in response to extrinsic information, right? So a migratory cell, again, we'll see that next time, tends to be attracted to a source of molecule of attractant, or instead repelled from a source of chemorepellent, for example, right? So there are often extrinsic factors and one of the important points we're going to have to think about is if you have an extrinsic information telling you this is where I need to go or this is what I need to run away from, then you need a relay. You need some mechanism to sense that, and you need some mechanism to convert this initial polarity or asymmetry into an intrinsic polarity. Okay, so we're going to have to think about those things. And finally, and I'm going to just touch briefly on this, but that's very important, that's about the consequences. What does that mean for a cell to have asymmetries to be polarized in certain ways? So this lecture, you'll see, is departs a little bit more from the textbook than previous lectures. So I kind of purposefully made it a little bit more, from primary literature, so I'm going to tell you more than what's in the textbook and sort of different things, and closer to actual research. So here I'm just highlighting a few of the models that have been used to study polarities, to study asymmetries, and I don't need to read this, but mostly I'm going to tell you about the work on the early embryos of the worm Caenorhabditis elegans. So many of you may be already familiar with those because we have a few faculty in this department who work with Caenorhabditis elegans. How many of you eventually work in these labs? Are there anybody? Matt Brockman, Sevinch Erjan, David Fitch? No? Nobody? Okay. Well, you have good opportunities here to have hands-on experience working with these organisms. They are, pretty fantastic, and I'm going to tell you a whole lot about them. And the reason why I'm going to tell you a whole lot about them is because this is the model that was used to discover a key polarity pathway, including the so-called PAR complex. I'm going to tell you over and over again about this. And at the end of the lecture, we're going to see another example, more briefly, about using Drosophila models, especially focusing on neuroblasts and precursors for sensory organs, mostly to illustrate two things. One, the deep conservation of mechanisms involved in polarity. So many were discovered in C. elegans, but later found to be conserved, in flies and all the way to humans. But also to illustrate a case where asymmetric partitioning of an important molecular determinants help a cell determine that it's going to become a neuron. Right? So that's somehow the business end of certain asymmetrical divisions. So now let's look at the mechanism from a bit of a conceptual perspective. And we can break down the establishment of polarities and asymmetries and their consequences into these simple steps I'm going to highlight here. So imagine you start with a cell, and here depicted are some components that are important for the identity of the cell, say these fate determinants here, these red pills, and then the big blue pill is the nucleus with DNA, and then those green crescents, they are so-called polarity cues, so in other words, molecules that will distribute in the cell in an asymmetrical fashion. So the first step, yes? Yeah, so I'm going to give you more specific examples, but yeah, so the idea in this cartoon is to say that you have proteins and molecular systems that are dedicated to establishing polarity, right? So telling the front from the back, the left from the right, the top from the bottom, and so on, okay? But that's what they do. That's all they do. And then the idea is that when you have a fate determinant, so a protein which alone does not know where to go, but will impose an identity, for example, or block an alternative identity. So the idea is to couple these two, right? So well, that's what I'm about to explain to you, actually, right? So the idea is that you start with a so-called symmetry breaking event, and like I said, this is often extrinsic to the cell. There are some cases of stochastic polarization, but in most biological systems, the symmetry breaking event is deterministic, so it's reproducible, if you will, and it comes from the outside. And I'm going to give you a few examples. Then you have a relay where polarity is established, and what I mean by that is those polarity cues or polarized molecules, they gather on one side of the cell, okay, in 3D. Okay, so one pole, if you will. Now, this can cause so-called segregation of determinants. So in this cartoon, it's represented as segregating in the opposite direction, right? So now you have this coupling I was mentioning. The polarity proteins, they respond to the extrinsic cue, they create this initial polarity, and then the determinant responds to that to position itself. Okay, so that we're going to see a couple examples of that. And then finally, in the case of an asymmetric division, that the asymmetric divisions or asymmetric partitioning of fate determinants are essential in embryonic development and other situations. So again, if you think about it, the last thing you need to couple with this polarity is the spindle, right? The positioning of the spindle. So that's what's showed here. And if you position the spindle on the same axis as the polarity cues and the fate determinants here, and here's the DNA, and I guess, so this represents kind of a metaphase plate. So the position of the spindle at the metaphase usually determines where cytokinesis is going to happen. So, right, so I know you haven't had the lecture on cell divisions yet, but cytokinesis is when the cell eventually pinches off and you actually have now two cells, two separate cells. So indeed, once you've positioned the spindle, when cytokinesis happens here, now you can have two daughter cells. And in this cartoon, there's a clear asymmetry in the sense that there's a small daughter cell and a big daughter cell. And when you have this kind of difference in 2 dimensions, that means in volume, this guy is probably about 3 times bigger. And then, as depicted here, only this daughter cell has inherited the fate determinant. So we'll see an example. If that fate determinant is telling, okay, now you're becoming a neuron, versus the absence of the fate determinant says you're remaining a stem cell, there you go. Now you have a division which has been asymmetrical by design, or I don't want to say design, by, the mechanism makes it asymmetrical, and the outcome is that you have 2 different cell identities, right? It's very important when you think about it. Okay, so let's look at the more specific examples of C. elegans. Oops, come on. So this is the C. elegans worm. How many of you have seen those guys? So you have, right? So you know some of the key advantages of those organisms for experimental research, right? So in a nutshell, this is the adult. It's extremely simple. There's about 1,000 cells. And as you can tell, it's very transparent. So by simple light microscopy, you can see individual cells. And people have traced all the lineages, all the cell divisions, leading to the formation of these adults from the fertilized egg. So it's been a boon for cell biology in an actual living organism. And people have studied quite a bit what happens in the germline. So there are still people studying the germline of C. elegans in this department, like Chris Gonzalez and Fabio Piano. The Fitch studies the tail tip of the males, and then there are Savage studies. The dosage compensation and genomics, and you have even more people studying C. elegans at the medical school. So it's really a workhorse of experimental research throughout the world, but in particular at NYU, actually. So the germline, let's just take a quick look at the germline, which in these hermaphroditic worms occupies most of the organism, right? So if you zoom, the germline, you have these two arms, so that would be the vulva. So they are hermaphroditic. That means at some point they can make sperm, but they also make eggs, okay? And they exchange sperm, so they can keep the sperm of another individual in this so-called spermatheca, or sperm storage system. And here is the gonad. So that's the now the female gonad, I should say, which produces oocytes. And the whole idea is that you have here a stem cell niche in some ways, and stem cells divide, germline stem cells. They divide, so they constantly produce new cells, okay? And then these new cells undergo meiosis, right? Hopefully, as you've seen that before, during reproduction in the germline, you have to have meiosis. So you have to divide the genetic material in two, have recombinations between paternal and maternal chromosomes, and so on, okay? And eventually, you produce an oocyte here, which will enter, which will get in contact with the sperm, and now that's fertilization. So the very first steps of embryonic development here, they do happen in the mother, the very first few divisions, and these features have rendered the system very tractable, very amenable to imaging, and so on. Now, I want to emphasize one thing from the get-go, is that by design here, for what happens in the earliest steps of development, you already have an extrinsic symmetry breaking event. And that is the position, so the cartoon maybe doesn't really do it justice, but in reality what happens is the oocyte, the mature oocyte, come from this ovary, and they meet, so the sperm comes from one side systematically. So the sperm entry point is a first symmetry breaking event, right? And I'm going to spend quite a few slides explaining molecularly what the sperm brings, at least one idea, and we're going to see the consequences for establishing polarity. Okay, so now we're going to watch this movie of the early embryo developing, and I want you to pay close attention because the movie spans almost the entire embryogenesis. But we're going to focus on the very first steps, like the first division, the first couple divisions. Might actually, okay. I'm gonna pause it. There you go. So you see, this is the first cleavage. I paused it on purpose because it goes too fast. So, and what I'm going to tell you is that the sperm entered here. Okay, so you see the first division. That's the cytokinesis furrow. These are the two daughter nuclei. What do you notice? It's very asymmetrical. Okay, so there's a big cell here, a small cell here, and that's just the first division. And this pattern is going to happen over and over again. Then it goes too fast, like to see anything. So normally these kinds of movies should mesmerize you and make you want to take developmental biology next semester with me, for example. But you don't have to. Or gastrulation happened. Yeah, in the worm, gastrulation is a little underwhelming. So what you were seeing was elongation and tail formation. And then the animal twitches. So you can actually, when you see all this twitching, you can appreciate that even before hatching, right, the larva is still in the eggshell. So that means it already has all the nervous system and the neuromuscular junctions in place because it's actively twitching. Okay, so let's look at stills because that's going to be a bit easier. So you have the egg. Initially, it's not really polarized, actually, right? So that polarity, as we'll see, emerges as a consequence of fertilization, but very quickly it leads to this asymmetric cell division. So a nuance between the two terms could be polarity is within a cell, you have an anterior side and a posterior side, and then the asymmetry is a result of that polarity. You have a small daughter cell and a big daughter cell. Okay, but you can use the two terms interchangeably, in my opinion. Right, so you have these two, and they have different names because people have been able to watch these worms and track the fate of these individual cells, and because it's extremely stereotypical, so it happens in the same way from one worm to the next. You can always recognize so-called AB versus P1, which is a small cell. Then AB divides again, as well as P1, and this division here is also asymmetrical. And then P2 will divide here, and that's also an asymmetrical division, and so on and so forth. Okay? So you can find these basic concepts in this particular case, right? So you start with a polarized cell. The polarity comes from the fertilization, and just after division, now you've already created two different cells, at least size-wise. We haven't seen yet more differences, more asymmetries, and I'm going to tell you about that. So, but sticking to C. elegans, one of the key points is, like I was saying, the divisions are stereotypical, so people could reconstruct these ontogenetic trees. So what does that mean? That means the first cell, the zygote, here divided producing these two, then P1 divided producing these two, and so on and so forth. And at some point, tissues become recognizable, right? Be it morphologically or using molecular markers, so genes, proteins expressed specifically in neurons, in the pharynx, which is what the worm uses to eat, or the germline here. And this tree is invariant, right? So that's an amazing feature. You have few cells making up every embryo, and these cells, they follow exactly this program from one worm to the next. So invariably, those cells here called Z2 and Z3, they come from P4, which comes from P3, which comes from P2, and so on, and they are the ones forming the germline. And that principle is true for all these cell identities showed here, right? And so this is also true in ascidians, the animals we study in my lab, and we think it's a property of embryos developing with few cells, where everything is hardwired. There's no room for chance, for variability. Everything follows a very strict stereotypical plan. So one question now becomes, if each one of these divisions, especially in this lineage here, leading up to the germline, is asymmetrical, and those cells, they acquire a specific identity, how come? What are the fate determinants, right? So this is a beautiful case to study this, to study how asymmetric cell divisions, which are emerging from an initial polarity, how can they lead to the production of distinct cell identities, and what are the fate determinants? Okay, so all these questions are somewhat connected. So we're going to address them, asking about the polarity cues and the fate determinants, at least to some extent. So in this slide, what I'm showing you is actually unpacking what happens initially, right? Because in the movie and in the previous slide, I jumped right away between the zygote, from the zygote, fertilized egg, to the two-cell embryo. But you have to now think, okay, well, wait a minute. Something has got to happen in between the symmetry breaking event and the actual division, right? So that first cell cycle, what happens in the zygote just after fertilization is going to be essential to set up the polarity and the subsequent asymmetry, okay? So this is what these images are showing are still. So you have here fertilization just happened, and here what you have with the marked with this O is the so-called pronucleus of the oocyte. So what is that? You remember those cells, they come from Hey. Oh. Sorry about that. Actually, never mind. Don't leave. Okay. Let's not get distracted. So the whole idea is that these cells come from meiosis, so they each have half a genome. So now you have, from the standpoint of that particular zygote, this is now the maternal genome, haploid, and this is the paternal genome, haploid, and it comes from the sperm. So the first thing, at least from a genomic perspective, is that those two have to come together and basically create the nucleus, reconstitute the diploid genome of the zygote, right? So that's what you see here. So there's quite a bit of activity. There are cortical contractions, and then the two pronuclei, they end up meeting. They tend to meet closer to this side. And then eventually you have one nucleus, which is now the zygote nucleus that has both genomes, it's diploid. And itself, now it can undergo replication and enter mitosis. Okay, all of this has to happen in that first interphase. Okay, and indeed, and now you have the beginning of mitosis and then the two cells. Okay, so in the meantime, there's a lot happening here at the cortex, and here you see you have this so-called pseudo-cleavage. That means there are these cortical contractions, but no divisions yet, right? You don't want to separate the paternal pronucleus from the maternal pronucleus. They have to merge first, okay? So let's see what happens. So one idea, though, is that by that stage, when the cell is about to divide, if you think back about the cartoons, like I'm showing you here, now you should already have, right, so here the metaphase plate is positioned, and it's positioned along this main axis. That's what you want, okay? So the fate determinant should be already positioned on either side, well, on one or the other side, okay? So now let me show you this movie. This movie is from a paper published now 10 years ago. And when it came out, actually I incorporated it very quickly in this lecture because this was a big game changer in this field. Actually, I'm thinking of creating a new lecture just about this. Have you heard about liquid phase transitions in cell biology? Yeah. So this is a very emerging, boiling field. So it's the idea that you have. So in physics, phase transitions is when you go, for example, the most common one is from water to ice, right? But you can have so-called liquid-liquid phase transitions where two liquids, so two different molecules or families of molecules of different properties can make separate phases, but both are liquid, okay? It's like oil in water. It's both liquid, but they separate, okay? So here, what you're going to see is so-called P granules. They've been known in C. elegans for a long time, and as you'll see, they will become polarized. They will get enriched on one side of the cell. So that's the main point here, actually. But what's beautiful about this movie is that if you focus on any given particle, some of them, you see them move, actually. But some of them, so now you see the asymmetry is getting quite obvious. And this was all during the first interphase. So now you have this little dance of the two pronuclei. Then the one nucleus goes back to the middle, and it doesn't go all the way. But if I play it again, so you have two kinds of mechanisms. Some particles move, but other particles, they just seem to vanish. And others are created de novo. And actually, pre-existing particles tend to increase. So here the polarity is a bit complex, and I'm not going to go into the details in this course, but is that the molecule, this GFP pGOL1 and some others that are part of these P granules, they tend to coalesce and so-called phase separate. So they become distinct from the cytoplasm. But their propensity to do that is higher in the back than in the front. In the prospective P1 than in the prospective AB cell. Okay, so now you have an asymmetry. You have asymmetrical distribution or polarized distribution of these P granules. Okay, so and this was known for a long time before this phase transition business came out. And the idea is that initially, so in the early fertilized egg, you have P granules all over. And then during that first interphase, they tend to accumulate on one side, and then there's a division. So after that division, those P granules, they are present in P1, but not so much in AB. Okay, so now you have asymmetrical partitioning of a potential fate determinant. I haven't showed you anything about this, but that's one idea. So it's true whether you do an immunostaining for some components or you look at this GFP-Pi1, which is one of the markers. So in live embryos, you can see it segregate. And you can see it segregate in the first division, but you can see it segregate in every subsequent division of the P1 cell. Right? So this is what this slide is illustrating. Now you see the embryo has a few more cells, and those P granules accumulate in this most posterior cell here, and later on only in this cell. And actually what people determine is that they always go to the most posterior cell, they wind up in the germline. So they are part, actually, of the molecular program to specify the cell to become a germline. And this is something that is inherited from the mother in some ways, because those components are present in the egg even before fertilization. Okay, so but they need to be asymmetrically partitioned to then influence the identity of cells down the road. Okay, so it's very important for the worms to be able to do that, and there are many examples of such asymmetrical distribution of fate determinants in worms and in other organisms. You have a question? No? Okay. All right, so why am I telling you all of this? Because those were wonderful assays to evaluate polarity and asymmetric cell divisions. And what the field has been pioneering some time ago now is to use this ability to look at C. elegans embryos and look at mitotic spindles here. So that would be for the second division. So this would be one spindle, this would be another spindle. You see the orientations are different. And the partitioning of the P granules. And then look for mutants and identify mutants in which these simple processes, simple to observe, are altered. Okay? So those were really pioneering studies. And here, for example, is one of the first mutants called PAR1 for no partitioning or partition defective, so no partitioning of those P granules. Sorry. So what you see here is that the spindles are formed normally, but the granules do not get polarized. Okay, and so it's one of them. And then people identified many mutants, which when compared to the wild type, you had abnormal partitioning of the P granules, and sometimes you had a normal mitotic spindle orientation, like in PAR1, but in other cases, here you can see, in this one, for example, normally the mitotic spindle orients along the anterior-posterior axis, and this one is oriented in the other direction. In this one, both spindles are oriented in the anterior-posterior axis, and the P granules, they become asymmetrically partitioned, but in the wrong cell, and so on and so forth. Okay, so those were really essential discoveries for this so-called PAR complex or PAR system, which has been named after those. But when you do forward genetics, so this was some kind of mutagenesis screen, right? So you induce random mutations and screen for mutants. So you know there's a phenotype, but you don't know the gene. You don't know the molecule, right? So that's the next question. What are the molecules? How does it work in the cells to achieve this? And by this, I mean positioning the determinants and positioning the spindle. Okay, that's what I'm going to tell you about. All right, so the PAR protein, so of course, since then they've been cloned, the genes have been cloned, identified, and the PAR proteins are extensively characterized. So we know quite a bit about them. So if you do an immunostaining for 2 of those, what's really striking, and now this is the C. elegans embryo before the first division, right? So during that period where I told you the embryo already has to be polarized. It already has to know intrinsically anterior from posterior, but it's not divided yet. Right? The P granules may already be partitioned, but there's not been a division yet. And of course, there's already accumulation of this protein PAR3 in the anterior cortex and PAR2 in the posterior cortex. So those proteins, they have a polarized distribution, and they tend to accumulate at the cortex. So by cortex, I mean the cytoplasmic layer that's just underneath the plasma membrane. Okay? And then when you look later, this polarity it persists. So PAC1 is another kind of molecule, but PAR3 here, you see, accumulates on the membrane that's facing the outside of the embryo, whereas this protein here accumulates at the cell-cell junction. So you have this polarity which is established early and persists through development. So I'm going to tell you more about this complex and the so-called PAR system, where you have, of all the mutants that were identified initially, now they've been classified in anterior versus posterior versus throughout. And simply put, in C. elegans, you have this PAR complex. So this one is extremely important. That's the one I'm going to focus on the most because it's very conserved, as we'll see. It works in polarity in worms, in flies, in humans, and so on. Okay? And as we'll see, it's a complex of proteins that contains PAR3, PAR6, as well as this kinase, so regulation of phosphorylation, and this CDC42. We've seen this CDC42 before, do you remember? In the axon lecture?
Actin.
Actin, yeah, what does it do?
It's a small GTPase.
That's a small GTPase, that's right. That's good. And no, that's good. And it can activate up to 3, promote actin filament elongation, de novo formation, and so on. And so you have components that go in the front, components that go in the back. Now, if I try to engage you again, what do you think could be happening? When you want to distinguish 2 different sides and keep them apart, like, left and right, front and back, Republicans and Democrats, what do they have to do? They have to fight. And that's what we're going to see here, that these asymmetries, they involve a lot of antagonistic activities between the 2 sides. And I highlighted the kinase because this is going to have a major role to play. Now, this slide is a bit scary. I'm not going to ask you to memorize it, but I just want to use it to illustrate some key processes, right? So again, these proteins have been characterized extensively. They tend to be, like many other proteins, they have different kinds of domains. And what I want to emphasize is that there's a lot of protein-protein interaction, especially within the PAR, the proteins that form the so-called PAR complex. So, for example, PAR6 here, you see can bind PAR3, can bind APKC. It's true for 6A and 6B. PAR3 can bind pretty much all of them. Well, it can bind itself, APKC here and PAR6 here. And APKC can also bind the PAR and has this kinase activity. Right, so you have everything you need to have proteins interacting with each other and eventually providing kinase activity in the PAR complex. This table here illustrates the conservation. So like I was saying, the PAR complex is extremely conserved. So you have PAR3 in C. elegans. It's been identified and called bazooka in the flies. Don't ask me why. PAR6 because it was first found in PAR6 in the worms, and so on and so forth. And they also, so this is a description of the proteins. And when people study their distribution, indeed, the simple view is that you have the PAR complex, so PAR3, APKC, PAR6 binding to each other, and they tend to localize in the anterior side of the egg. Come on. What the? This is embarrassing. I'm going to put you on airflow. Sorry about that. Right, so as you see here, you have PAR1 and PAR2. So those are more specific to C. elegans, especially PAR2. Here, there's no homologue that was found in other species, really, but it's part of the C. elegans machinery. And those guys, they tend to localize on the posterior side. And then you have some other proteins which are not really localized. They are found throughout the cytoplasm. And as we'll see, they contribute to the crosstalk between the two. So how does this crosstalk works? I'm going to show you in this animation of the cartoon. The principle here is described in an epithelial cell, but it applies, across the board. So here is how it goes. So imagine you start with PAR3, and it happens at the cortex. So I showed you PAR3 tends to be an anterior localized protein. So when it wanders in the posterior side, somehow you have to have a mechanism to displace it. Okay? That mechanism involves PAR1. So that's the first step here. And PAR1, we've seen it. It tends to localize at the back. It will phosphorylate PAR3, and that phosphorylation causes it to leave the membrane. And now it's bound to PAR5. So there's basically an active removal of PAR3 from the membrane, from the cortex, in the back. That involves PAR1. Now, on the other side, there's no PAR1, and actually the antagonism, and one reason why there's no PAR1, it's because it's being phosphorylated by APKC PAR6, which is positioned there, and here as it's showed is by binding to CDC42, but also PAR3 as a matter of fact. So this leads to the same principle for PAR1. So now you see you already have an antagonism between this PAR complex and PAR1. And eventually to finish, once this goes back to the anterior side, it can return to the cortex, and if it goes this way, it's stabilized by its interaction with the APKC PAR6 and CDC42. And same thing for PAR1. So that helps explain the maintenance mostly. Okay, if you assume that the cell already has PAR1 on one side and the PAR complex on the other, this mechanism explains the maintenance of that partitioning. Okay, you see that? But it doesn't explain an important thing, which is what? Which is how it's established in the first place, right? And in particular, what did I tell you? What establishes the asymmetry in the first place? Fertilization. Sperm entry on one side. So a key question you might have at this point, I know you're dying to know that, I can see that on your faces, is how on earth does sperm entry trigger the partitioning of these components to the anterior versus the posterior? And that's what we're going to see. So in this movie, we're going to be looking at GFP PAR6 as a first entry point to understanding the dynamics. In other words, how is this asymmetry established? So what's the phenomenology to begin with? So it's at the core, and here's the pseudo. Okay, so this may not be the most striking thing, but it's pretty clear actually what happens from this movie. So most of the signal you're seeing is at the cortex. Okay, And the egg, you don't see the outline very well, but it basically covers the entire screen. So initially what you have is low level of PAR6 everywhere. Okay? And then the dynamics, simply put, is that it becomes excluded and somehow shifts anteriorly. Okay? So that's the first insight. The establishment of this asymmetry is the PAR complex, PAR6 for example, tends to be at the cortex, just shifts anteriorly. So in stills, indeed, this is how the movie looks. The sperm entry point or the would be here. PAR6 is initially almost everywhere and then eventually cleared from the back and accumulates anteriorly. And if you look at this kymograph, so remember the kymograph is a way to look at position in one spatial dimension. So here anterior posterior in the x-axis over time. So early you have broad distribution of PAR6 GFP or GFP PAR6, and then over time it restricts to the anterior, so it tends to accumulate in the anterior. It's being cleared from the back. Okay, so one question we now have to have is how, right? So again, PAR6 tends to be everywhere. It's true also for PAR3. It's true for the PAR complex. Okay, so there's no polarity initially. And that polarity is established by removing the PAR complex from the cortex in the back and actually replacing it with PAR2, and it has to do with sperm entry. Okay? Now, what in cells can move things around? Especially at the cortex. What kind of cytoskeletal elements are at the cortex? 2 lectures ago. Actin microfilaments. What kind of molecular motor works on actin? Myosins. Let's look at myosin. Right, so you see myosin, and you see, so this movie was kind of a landmark. So this is non-muscle myosin light chain fused to GFP in the early C. elegans embryo by Ed Monroe. So initially you see a whole lot of activity, pseudocleavages there, and eventually you see a lot of myosin also winding up in the front. But I want you to pay attention to the dynamics here. See these foci, and there's some kind of pulsatile activity. Okay? But so there are essentially 2 things to see from this movie. One that is quite subtle is what happens very locally, this sort of pulsatile contractility. And then, but at the level of the whole cell, what do you see? You see the myosin is also cleared from the back and moving anteriorly. Okay? So again, in still, this basically, it starts broadly. You have these foci, and then you have kind of a network of myosin staining, and then eventually, here's the sperm entry point depicted here, and eventually it clears from the back as well, winds up accumulating in the anterior. Same for actin filaments, okay, which makes sense because they tend, the contractility has a mechanical leverage only if they are bound to actin filaments. Okay, so basically it's like you have a drag of the entire actomyosin system towards the anterior, cleared from the back, right? And the interpretation of these foci is that if you have a network of actin filaments and you have myosin heads, so in this case, it's not the myosin that will transport a particular cargo, it's the kind of myosin that will bind with heads, motor heads on either side to bind to 2 different filaments and eventually constrict them. So one idea, which has since been observed in many systems, is that if you have a meshwork and if you have actomyosin, you tend to have accumulation in foci in this way, okay? But the contractility is spread everywhere. So what regulates myosin contractility? Yes, there's a kinase, and upstream of the kinase. Well, a small GTPase is always is kind of a go-to answer, right? Indeed. Remember the most important slide of the entire course. The raw GTPA, the small GTPAs, they control almost everything the cytoskeleton does, including actomyosin contractility. So this branch here starts from rho, one of the founding members of the family, activates this ROCK effector, and eventually, indeed, a kinase, myosin light chain kinase, and this is how myosin gets activated downstream of rho. And indeed, in the early C. elegans embryo, if you look for myosin light chain, phosphomyosin light chain, so that's a sign of active myosin, you see it at the cortex, initially throughout. This is also where the non-muscle myosin GFP accumulates. But in the Rho1 mutant, no accumulation of the cortex of either myosin light chain 1P, immunostaining, or NMY. So that's regulated by Rho1. Okay, because what are we after? I know I'm giving you a lot of information, and I can tell from your faces that I don't want to lose you here. We are still after the connection between the asymmetry of the PAR proteins and the fertilization. That's where we're going. Okay, so bear with me. What regulates small GTPases? Most important slide of the course. GEFs and GAPs. Okay, so this must become, by the time you're done with this course, I want you to have a Pavlovian reflex almost. When you hear small GTPases, you must be thinking GEFs and GAPs. Like, what's the gap? What's the gap? So in this case, indeed, we've just seen that, right? The asymmetry of PAR6 mirrors that of myosin contractility, which is controlled by Rho. So somehow the GEFs and the GAPs controlling Rho may have a role to play, may connect this contractility, this polarized contractility, with the entry of the sperm. And that's where we're going. So indeed, so the GEF is ACT2 here. It's an activator because it promotes GTP exchange. The GAP is CYK4. And let's see how this works. So in the ACT2 mutant, you get no activation. So far, so good. In the CYK4 mutant, you get activation everywhere. So far, so good. That doesn't tell us where the asymmetry is. I'm just going to go a little bit more quickly here. So, yeah, this one is not as clear, but the key point I want you to get from this is I'm telling you about Rho activity, the gaps, and the contractility, but I have not connected that to the PAR localization yet, right? So that's what this slide is showing. Now you have, if now you're looking at PAR6 GFP, but you're inhibiting Rho activity, or you're inhibiting the gap, you don't have asymmetry in either case, okay? So what this is showing us is that the asymmetric distribution of PAR6, that gradual removal from the posterior cortex, requires both activation of Rho and inhibition of Rho. It basically requires spatially controlled contractility. So still, where does the polarity come from? One thing I have not showed you, and it's still a little bit controversial, but the model was very appealing, and as a matter of fact, it was published in Science. I've not showed you where the proteins are: the ACT, the activator, the GEF, or the CYK4, the GAP, the inhibitor of Rho. ACT is everywhere. So there's basically constitutive, and you might expect that, right? Because when I showed you the myosin movie, initially, there's activity everywhere. So you have to invoke activities everywhere. As a matter of fact, I could ask you a question in the exam, showing you the activity and asking you, where is the GEF? And you should be able to say, everywhere, right? because without the gap, there's not going to be activity. But the gap is the key. And indeed, what these slides are showing is that if you label the sperm here with this sperm marker and CYK4, it's in the sperm. So that's a beautiful system. Now you have, indeed, the sperm, by fertilizing the oocyte, it's bringing the DNA, it's bringing the centrosome. Yes? Yes. The CYK4 is the gap. So that's the inhibitor of RO1. Right? So the idea here is that the sperm is basically bringing very locally an inhibitor of Rho activity. Okay? And now that's going to trigger, the onset of polarization. And in this experiment, what they did is if they use a mutant egg for CYK4, but a wild-type sperm, then they can get polarity. Okay? So the function of CYK4 is important in the sperm. So now let's go back to discussing a little bit of a model and the one idea. So there are many parts here, many moving parts. What I just told you is that the sperm, so fertilization, brings the gap, which starts to locally inhibit Rho A, whereas the activator, the GEF, is present everywhere. So now that helps, with the forward move, the anterograde movement of this myosin contractility, and with the PAR complex essentially following. So now, and these two things feed into each other, right? So, in other words, Rho A becomes displaced anteriorly. It drags the PAR complex, which promotes Rho A activity more anteriorly, etc., etc. On top of that, you have this other branch, which I have not showed you any data about, that fertilization does not only bring the gap, it also brings the sperm. So MTOC, we've seen it last time, is microtubule organizing center. It brings a centrosome, okay, which is very important for the subsequent cell divisions. And the centrosome allows for localization of the PAR2 protein at the posterior. So now you have almost a double kinds of negative inputs on the PAR complex, which is going to end up anteriorly, because not only are you inhibiting its positioning at the back through the effect of myosin, you're also inhibiting its positioning by positioning PAR2. Okay, so these other cartoons are essentially trying to illustrate that. Especially this one is probably the clearer, right? So you have this negative effect on a Rho activity, which has a positive effect on positioning PAR, so moving it anteriorly. And at the same time, the centrosome helps position PAR2, which also antagonizes the positioning of the PAR complex. Okay, so that's how you have the relay between the initial symmetry breaking event, sperm entry, and the intrinsic polarity, PAR complex anteriorly, PAR2 in the back. And we've seen that once you start establishing this asymmetry, the antagonism between the two, so these two negative arrows here, these two, they basically maintain and reinforce the asymmetry. Okay? So I'm done with the PAR complex at this point. Oh, wow. It took a long time. So let me move forward a little bit, just saying that everything that's been dissected regarding the PAR complex, dissected in great details in the C. elegans early embryo, turned out to be largely true in many other systems. So the PAR complex, so I mean PAR3, PAR6, CDC42A, PKC, they are involved in polarizing the Drosophila egg, epithelia, neuroblasts, as we might see briefly, as well as crawling cells, neurons, and so on. Now, we have 10 more minutes to talk about how to convert this polarity of the cortex into an asymmetric cell division. right? So what do you need to get an asymmetric self-division? To position the spindle. Okay, so let's look at that. First, I'm going to show you this gorgeous movie. So now this is, tubulin is labeled. And this is the early cell against embryo around the metaphase plate. So you have this very nice dance of the spindle. And because it's a spindle, you have two empty OCs, two centrosomes on either side. And you have essentially two kinds of microtubules. Some that form the spindle, and among those you have those that interact with the chromosomes and those that interact with each other, especially with the kinesin 5, and help, build the spindle and position it. And then you have the astral microtubules. So if you look at these guys, so wherever there are arrows, so what do you see? Dynamic instability. We've talked about this on Monday. Microtubules grow, microtubules retract. Catastrophes happen. What stabilizes them? Interaction with target proteins, where? At the cortex. So now you start having a mechanism. How are you going to position that spindle? How are you going to position that spindle in a way that the division is going to happen with regards to the polarity of the cortex, right? So you need this interaction between the microtubule and the cortex. And that's affected by those astral microtubules and how they will interact with the cortex, which is different in the front, in the anterior part of the egg or the posterior part of the egg. So here you have a very direct connection potentially between the polarity of the cortex and the positioning of the spindle. So let's look into that. And essentially what you'll want to do is the following, is modulating forces of interactions between those astral microtubules and the cortex in a way that the spindle has to be positioned in two ways. The first one is along this axis, this anterior posterior axis, and then the other way is slightly shifted towards the posterior, because remember that division is asymmetrical. Okay, so let's see how this is achieved. So this is a cartoon showing a model for how the microtubule interact with the cortex in details. And so you have these complex of proteins, and so you have these G-alpha proteins interacting with the membrane. It's actually anchored at the membrane. It has a lipid modification. This linker system, GPR12, Lin5, I don't need you to memorize this. If I want to ask you questions, I'll give you the information, and I want you to be able to think about it. And here there's this. Dynein. Does that ring a bell? Yes, a molecular motor. Why a dynein and not a kinesin? Dynein will walk towards the negative end, right? So now that topology, I would like you to understand it. Because what I told you on Monday is that microtubules, they grow with the plus end towards the cortex. So if you want to be able to pull on the microtubule, to exert force on the microtubule, and you know the plus end is here, what you want is two things. You want a motor that's going to tend to walk towards the inside, so towards the negative end, so that has to be a dynein. And if you want any traction, what you need is an anchor. So you can't have that dynein just move. You need to anchor that dynein to the cortex. And that's what happens here. Okay? So, and this system is actually very conserved. So in C. elegans, you have G-alpha, GPR12, Lin5. The names are here. And you have the equivalent in Drosophila. You have the equivalent in vertebrates. This is a basic mechanism to anchor spindles with the cortex and position them with regards to the orientation of the cells. Okay? Now, of course, and I know I've told you a lot of things in this lecture. So I think I'm not going to go into the fly stuff because that would be too much and we're running out of time, but that's okay. But the ultimate thing I want to finish with is how do you connect this anchor? Because what I'm telling you here is you're only anchoring the cortex, the spindle to the cortex. We have not yet answered the final question, which is how do you position it once you've anchored it, right? So there is some answer in this last slide I'm going to talk about today. So, well, I'm sorry, we're not going to go into the fly stuff, but I'd rather try to explain better the C. elegans and focus on the C. elegans part. So that means I won't hold you responsible for the fly stuff in the exams. All right, so here this is how it works, right? So we've seen establishment of this polarity at the cortex, okay? And eventually what we want is asymmetrical positioning of the spindle by differential pulling. So now if you think about it from a rather simple mechanical perspective, to orient the spindle, you want to have a ring, kind of an area in the cortex where there's very little pulling. Okay, so that's this area here. Okay, because if you imagine you have a belt where there's very little interaction, by default the spindle, instead of orienting itself in this way, for example, it will orient itself in this way, okay, along the anterior posterior axis. So that's one answer, and that's affected by this protein LET99, whose homologue is called DEPDC1, and it turns out we studied this in my lab, but that was anyway. And then the other part is to have slightly more pulling in the back than in the front, okay? Because that will allow the system to position the spindle slightly offset from the middle. Okay, and this is more or less directly affected by the part complex. So for example, if you look at this here, you have this activity of this kinase, and I don't need you to memorize all these steps, but just to illustrate the connection. You have basically positive and negative interactions between kinases, which are themselves polarized, and those directly impinge on the anchor here, allowing an increased force on the back and therefore slightly pulling the spindle towards the posterior. Right? So I'm going to stop here. Actually, I'm going to show you this last point of the fly. I'm not going to hold you responsible, just to show you this last movie. So this is a neuroblast, a neuron, actually a sensory organ progenitor. And what the movie is going to show you is asymmetric distribution of this PON, partner of numb, which is an important determinant for the GMC, the neuron progenitor. So you see the division happens, PON gets accumulated in one place, especially at the metaphase, division happens, and now all the protein is in one of the daughter cell. Okay? And now that daughter cell is endowed with the ability to become a neuron. All right, let's stop here.